I'd like to take a moment to introduce our presenter, Dr. Hayes. John is an award-winning leader of a transdisciplinary research program here at Penn State that applies sensory and consumer science to a wide range of topics, including, but not limited to, ingestive behavior, flavor perception, individual differences, and user-centered drug delivery optimization. Since joining the Faculty of Food Science in 2009 and becoming director of the Sensory Evaluation Center, John has led numerous studies using psychophysics to study chemosensation, genetics, food choice, and more. During the COVID-19 pandemic, Dr. Hayes collaborated with colleagues on a global survey to quantify the prevalence of loss of sensory function related to the virus. This eventually led to the Stop, Smell, Be Well public health awareness campaign and the distribution of coronavirus scent cards across our campus. John earned both his bachelor's and master's degree from Cornell and went on to earn a graduate certificate and PhD at the University of Connecticut. He did his postdoctoral work at Brown University. John has published more than 140 peer-reviewed papers and is regularly cited in national and international media. In addition, John has mentored more than 25 trainees who have gone on to very successful careers in the food industry and in academia. So John, I'm going to turn the floor over to you.
Thanks so much for the introduction, Bob. And I am excited to talk to you today about a passion of mine, which is the science of food preferences. So let me see if I can get this to share. All right. So hopefully now you're all seeing that. So the formal title of our chat today is called The Science Behind Our Food Preferences. But the subtitle that I'd really like to dig into is I like it hot. Wait, not that hot. Yikes. Or in other words, we are going to talk using one example of chili peppers to explore all the different ways sensory and consumer scientists can both use people to study product and use product to study people in a bidirectional manner, because we do both of those in our research program here at Penn State and the training that we give to our students. And then at the end, I'll broaden it out a little bit beyond chili peppers to talk about some of the other work that we've done to try and increase the tasty food in the food supply. So we can start off by asking, what is sensory evaluation? And this is a very old definition that dates all the way back to the 1970s, used by the formal professional organization, by most food scientists, and also it shows up in many textbooks in sensory evaluation. And I really want you to focus on three words there. So the sensory evaluation is a scientific discipline used to evoke, measure, analyze, and interpret responses to products that we perceive through our senses. And so this is that one-directional relationship of using people to study product. But as I said, we can also go in the other direction. And it's really important to understand here that sensory evaluation is not the idea of being an expert taster. We're not Robert Parker rating things for the Wine Spectator to give it a score of 93. That's not what we do. Instead, we really democratize this process by we bring in dozens or even hundreds of individuals into the lab and collect data from them under very controlled conditions, and then use that quantitative data to make predictions about what foods people will like. So the fundamental principle here that distinguishes us from things like market research is we're not interested in branding, we're not interested in price, in the value proposition. We really don't care if you like Coke versus Pepsi. Instead, we want to know if Formula A tastes better than Formula B, because if I work at Pepsi, regardless of which formula I essentially decide to launch as a new product, it's going to have Pepsi on the can. So we're trying to tease apart the sensory differences that drive differences in liking from the overall product characteristics like you would in market research. And so we do all of our testing blind. And when I say blind, I mean this in the sense of a clinical trial is blinded. We don't actually use blindfolds, unlike this picture on the left, because frankly, the food goes everywhere. But it's really just to illustrate this idea that we've removed all that contextual information as much as we can to try and narrow in on the product itself as it's presented under controlled conditions. And so here on the right, we're seeing a photograph of one of our 12 taste test booths here in the Erickson Food Science Building. And this individual is evaluating wine samples that have been served on that tray in front of them. They get passed through the hatch in the top left corner. We ask them to rinse with water in between, and they answer questions on the computer. So we use digital data collection to facilitate both the experimental design and also the data analysis. So telling you to test sample 643 before you taste sample 721 and so on. And we use 3-digit blinding codes, for example, to then avoid that sort of information of this is Coke, this is Pepsi. And I'm just one member of our very large team here. So as you heard, I was hired in 2009. Professor Helene Hopfer was hired a few years later. And so the two of us, we make up the 2 tenure line faculty here in sensory and consumer science. We also have 2 affiliate faculty. We used to call these adjunct faculty, but we've changed the name. One of them is Dr. Sarah Kirkmeyer, and she has 30 plus years worth of experience in food industry and also the consumer products industry. And then Dr. Catherine Ola in Switzerland is also an affiliate faculty. We have a full-time staff member who helps us run the Sensory Evaluation Center. And then we have our research footprint that consists of 1 or 2 postdocs, 5 to 10 graduate students, and then every semester we employ almost 20 undergraduates to help us in the lab to run those taste tests. Because when you're running a taste test under controlled conditions for 100 people in a few hours, it takes a lot of labor. And sometimes we've had to adapt. And so we're always adjusting to the circumstances as we can. So for example, there on the bottom right, that's us testing during COVID, where we actually gave people product to take home and use it at home because our lab was closed for 15 months for safety reasons. So we still needed to collect those data, but so we had that trade-off of being able to collect data safely versus doing it in somebody's home. So some of the products that have either been worked on in our laboratory as part of experiential learning to give our students experience working with real products and not just research samples that have been made by our graduate students for their theses and dissertations, but also many of our alumni go on to very successful industry careers. So that includes things like all the Quaker granola bars. That includes things like Ocean Spray cranberry juice, the Boston Beer Company, which makes Truly. Also, more regional breweries like New Glarus in Wisconsin, very famous award-winning brewery there. Of course, we're Penn State, so we have to work in chocolate, both at Hershey's and at Mars Wrigley. But in the top right, you'll notice some non-food products, because many sensory and consumer scientists also get hired by fast-moving consumer goods companies, because the same methods that we use to evaluate foods can be evaluate other consumer products. In fact, most of the consumer products and most of the foods that you eat or use on a daily basis have probably been optimized by a sensory and consumer scientist. So now let's dive a little bit into the science. I know this is a really overwhelming figure, but I'll walk you through it very slowly. So over on the left side of the graph, what we can see is we can see 7 different receptors, and these are called trip channels. And these trip channels, it turns out most of them are temperature sensors. And so we can see over on the very bottom left of the plot, we're responding to very cold temperatures. On the right-hand side of that plot, we're responding to very high temperatures. And this work has really, our understanding in this has really been revolutionized over the last 20, 25 years. TRPV1, which is probably the most well-studied of all of these receptors, was first discovered in 1997. In fact, in 2001, that discovery earned those researchers the Nobel Prize. And these trip channels, it turns out, are actually heat pain sensors or cold pain sensors. So when I go and I have some mint or I have some menthol, like that cough drop that you might be using this time of year, that cooling sensation you get, your mouth is not actually cooling off. Instead, the chemicals in that product are activating one of these receptors that then is sending a signal to your brain that says either that's cooling or that's warming, even though the temperature of your mouth is not changing at all. And so, as I said, the best studied of these is TRPV1, which normally responds to a temperature of about 42 degrees Celsius, or for those of you that think in Fahrenheit, that's about 106 degrees Fahrenheit. So we know this: if I put your hand on a hot plate and turn up that hot plate and have you pull it off when it gets too hot, that's about the temperature that's going to happen. And that's because we're activating these TRPV1 receptors, both in the skin, but also the same thing would happen if I did it in the mouth. Well, the chili plant has involved a chemical called capsaicin that actually fits into that molecular thermometer, and it lowers the temperature, kind of like a thermostat, turning down a thermostat. And so now instead of activating at 42°C, it activates at 30 or 33°C. And again, if you don't think in Celsius, human body temperature is about 37°C, human mouth temperature is about 35 or 36°C. So now we've lowered the temperature at which this thermostat responds. And so just the sheer act of putting chili peppers into our mouth or putting capsaicin into our mouth will actually cause those burning sensations. And the reason the chili plant evolved this compound was because plants don't want to be eaten. But if you're a plant, you can't run away. So you will often come up with these secondary metabolites, as they're called, to prevent predation. And so in the case of mammals, we're not supposed to like spicy things. In fact, it's a highly aversive experience. Most mammals will not eat chili-containing products. This is why if you want to put cayenne pepper into your bird feeder, it'll keep the squirrels out. Now, interestingly, the evolution here gets a little quirky because the avian version, the bird version of the TRPV1 receptor, doesn't respond to capsaicin, which is again why we can put chili pepper into our bird seeder, because now when we put that bird seed in our bird feeder with chili peppers, the birds literally don't experience the heat while the squirrels will, or the raccoons will. And so this is an aversive response that generally we don't like. And in fact, we know this in young humans. And so these are pictures that a colleague of mine took of her young son. He liked candy. He had never had an atomic fireball. And his mom said, "Would you like to have an atomic fireball?" And he said, yes. And you can see that he was a little excited at first. And then over time, as that burn built, it was a really unpleasant sensation. And he was not enjoying it very much at all. And by the end, he was quite miserable. So why do we like to eat these things when they're aversive? And so that's one of the things that we try and figure out in our lab. And so I want to digress for just a minute, because you're probably familiar with the idea of market segmentation. So lots of times different market researchers will use demographics to try and split the market into these consumers versus those consumers. The people that are buying a Ford F-150 and the people that are buying a Prius are different consumers, so that you're going to target your marketing to each of them. And often researchers will come to us, or industrial partners will come to us with a very specific idea of who their consumer is. You know, it might be a 35-year-old woman who has 2 young elementary school age kids and is the primary household shopper. But I want you, as a thought experiment here, to think about a really precise demographic. So imagine a white male who's a highly successful businessman. They make more than $1 million a year. They're English by birth. They've been married and divorced. They have 2 adult children. They really love dogs, and they like to go skiing in the Alps. So you probably have a very specific image of this person in your head right now. In fact, sometimes demographics can leave us down, because it turns out that both King Charles and Ozzy Osbourne fit this very precise demographic profile. And yet it's hard to think of people that are very different. So we need to move beyond simple demographics. And instead, we can look at something that are known as psychographics. So we can look at sort of the motivations that these people have, rather than just a pure description of who they are demographically. And you're already familiar with this targeted market segmentation, because every single time you go to buy salsa, every brand of salsa out there, almost every brand, I shouldn't say every brand, almost every brand makes a mild, medium, and hot. And this is because if we make a one-size-fits-all food, then it won't appeal to everyone. Instead, it'll just appeal to a few people. So instead, if we make different products for different market segments, we can really tailor that product to make that smaller group of people happier. And so you can imagine a process where we bring people into the lab and we give them different salsa, and then we ask them on this 9-point scale to indicate how much they like this salsa. And so this scale was originally developed by the US Army in the 1950s for optimizing rations. And it's been used thousands and thousands of times since, in academia, in government, in food companies as a way to figure out what food products people like. And again, I'm not asking that one expert taster to come in and evaluate a product and tell me if it's good or not based on some platonic ideal they have of what salsa should be. Rather, I'm bringing 100 or 120 people in to the lab and saying, how much do you like this particular formulation tested blind? But I can also do something like just about right scale. You can think about this literally as Goldilocks and Three Bears. Is there not enough cilantro? Is there just about right cilantro? Is there too much cilantro? And so I can collect those data quantitatively and I can start to analyze it. So these are made up hypothetical data, but let's just imagine the data that we might get from this. So the market leader, we really want to have very, very thin tails, and we want to have lots of that big white section in the middle, because that means we're just about right. So now you can compare our market leader that maybe there's not quite enough cilantro in it for about a third of the population, and maybe about 15%, so there's way too much, but there's a big group of people in the middle that are pretty happy with it. Now consider our new prototype that we're evaluating. And we've overshot our marker. You can see that almost half of the participants say that there's too much cilantro, versus very few of our participants are saying that there's not enough. And then you could look at something like prototype 2, and here we've really hit the mark. So with prototype number 2, we have a few people that are annoyed that there's not enough cilantro, and then we have a few people that are annoyed there's too much, but most people fall right in the middle. And again, we want to bring in a specific demographic of users or a specific product profile of users. So if I'm testing medium salsa, I want to bring in people that usually buy medium salsa and not just all salsa consumers. And so you can see that again, prototype 1, too much cilantro, versus prototype 2, I'm actually doing a pretty good job. And so again, these are totally made up hypothetical data, but in practice, you can actually see this kind of targeted market segmentation where you're only going after that 10 or 15 or 20% of the market, but you're making those people really happy. So in fact, here's an example of a product that's out on the market that actually advertises that it has extra cilantro. So it's going to really appeal to those cilantro lovers, but not those cilantro haters among us. And so coming back to this question of psychographics, about 10 years ago, we did some research where we were asking, can personality traits actually predict what you like? And lo and behold, what we found, my graduate student, Dr. Nadia Burns, appropriately named for this project, what Nadia did was she asked this question of, does the personality trait of sensation seeking predict how much you like spicy food? And it does. And if you're not used to looking at human behavioral data, this is an incredibly strong relationship, much stronger than we typically see. If you're a physicist or a chemist, then maybe these data don't look so great. But to a human behavioral scientist, this is a really robust effect. And in fact, this is very specific to spicy food. We find that it's not just the people that are high in the personality trait of sensation seeking. There's no relationship between sensation seeking and how much you like fried chicken or how much you like a vanilla milkshake. So it really is something about that spiciness. And in fact, we find that liking predicts intake. So if you like something more, you're going to eat it more. But I also want you to notice in this bottom right corner, there's this big gaping hole. And so these are people that like a product, or rather, if you don't like a product, you will not eat it. Versus up in the top left, you occasionally get people that like something but don't eat it. You know, I might like a big juicy porterhouse steak and a big Cabernet Sauvignon, but that doesn't mean that I'm going to eat them every day, even if I like them very much. And so in science, we always want to make sure that we can replicate things. And so Nadia went out and got another 100 people to come into the lab. And what we were actually able to find is a little more complicated, but I'll walk you through it briefly. We looked at the personality trait of sensation seeking, and we found that this actually predicted, we actually made jelly samples. We made strawberry jelly in the lab, and we actually spiked them with a specific amount of capsaicin. Now recall that that's the chemical that makes hot peppers hot. And then we also asked people on a pencil and paper survey how much they liked a spicy meal and how much they liked the burn of a spicy meal. And what we found is the personality trait predicted all three of those, and then how much you liked those things in the lab predicted how much you ate. But what we also found was there were two other personality traits, sensitivity to reward and risk-taking, and they were related to each other. But we found that they predicted whether or not you eat spicy food, but not whether or not you like spicy food in the lab. So from this, we infer that there's actually two different reasons people tend to eat spicy food. One is because they truly do like that burn, but another is because maybe they like some of the social reward that comes from being able to eat spicy food. You know, you're out with your friends, you're eating those wings, and you don't want to chicken out. So maybe you respond to those social cues as well, as opposed to just how much you like the spicy food. So we can start to tease apart the various factors that are myriad in defining what we like to eat. So often you'll hear people talk about this idea that if you like food more, you'll eat food more, which raises the question of whether or not food can be too delicious. And in fact, study after study after study has found that there is a positive relationship between liking and intake. If you like that food more, you're probably going to eat it more, or you're going to select it more, or you're going to pick it more. That's been shown over decades. That's been shown across continents. However, there's a really important nuance to that. Do you remember a few slides back when I showed you that empty hole in the bottom right, if we actually look at the raw data? It's not that liking a food makes us eat more of it. Rather, if we don't like a food, we won't eat it. So these are data from the US Army from about 20 years ago, where they actually studied thousands of soldiers in the field and gave them rations and actually asked them how much they liked those rations and measured how much they ate of those rations. And so what we can see is on that 9-point scale that goes from dislike extremely to neutral at 5, all the way to like extremely at 9, moving from left to right. What we can find is as long as you're at neutral or above, you eat almost all of it. And so it is true that if you don't like something, you won't eat it. But on the flip side, just because you like something a lot, just liking it moderately, doesn't actually mean that you'll eat more of it. And this study went one step further, and now it said, okay, now we know how much you like it the first time you were offered it. But now the next time those soldiers were offered a choice of whether or not they wanted to pick the exact same meal to eat, what we actually find is a very different story. So even if I like something at a 9, as high as it can possibly go, I like that extremely. What we find is that those soldiers still only pick that food about half the time. So there really is an important nuance here that yes, the more like the food it is, the more often it's eaten, but that doesn't necessarily mean it's true that just because we like something, it's not necessarily possible to make food too delicious that we just lose control. And so that's an important thing to keep in mind when we're talking about human eating behavior as it relates to the pleasure of food. So coming back to chilies, here's some data that we collected recently in the Netherlands, where we actually took chicken bouillon and spiked it with different amounts of capsaicin. And you can see as we add more capsaicin, it burns more. But you'll also see that there's a difference there between two groups. And we've known, dating back at least to the early 1980s, that frequent consumers and infrequent consumers actually differ in how much burn they experience. So it really is true that you can build up a tolerance to chili peppers. And this is, or rather, I should say, we know that people differ. And it's been assumed that this is because we can build up a tolerance. And in fact, if we go and we look at their liking ratings, what we can find is we increase it. If you make it burn a little bit, both groups like it more than just plain chicken bouillon, because chicken bouillon is probably pretty boring by itself. But then when we make it really hot, what we find is that the frequent consumers still like it quite a bit, where now the infrequent consumers are not so happy with those, because they're kind of miserable right now. And so at least half a dozen studies have shown these kinds of effects, but that doesn't actually tell us, is it those people differed originally? Were they somehow different out of the gate? Or is there something about prior experience? Is it repeated exposure? So we asked exactly that question. And so we actually brought people we ran in a small randomized controlled trial where we randomized people either to a treatment group or to a control group. So the treatment group, they got a bitter chemical called SOA, and we asked them to use that as mouthwash twice a day for 14 days. And then we brought them into the lab on day 3 and day 17, or as it's labeled here, day 0 and day 14, because it was a 14-day exposure protocol. And so what we can find is that the burn of capsaicin, the burn of chili peppers, for the group that got a bitter mouthwash every single day, for twice a day, in fact, for 14 days, 28 exposures, that group didn't change in their burn at all. But then we actually found that the people that got a capsaicin mouthwash every single day. So these people got exposed to capsaicin about 28 times over the course of 2 weeks. We can actually see about a 30% reduction in how much burn. And this actually showed us that it's inducible by exposure. So this confirmed the speculation that's been around for decades that you really can build up a tolerance. So we wanted to take this further. I told you at the top that we like to use people to study product, but we also use product to study people. So we actually did tongue biopsies on the people, and we took microsurgical scissors and actually lopped off tiny bits of their taste buds. And I can tell you, I've had this done to me personally, and it really doesn't hurt at all. In fact, it's far less painful than biting your tongue because the scissors are really sharp and the amount of tissue that we take is really, really small. So then we went and we tried to see if we could find any differences in the amount of the messenger RNA for the TRPV1 receptor, which recall. All right, hopefully I'm back. My Wi-Fi dropped for a second there. So I'm going to keep going and hope that you can still hear me. So we were unable to find any difference whatsoever in the biology. So we found the behavior changed, but the biology didn't. So this is a great example of how we're really pushing the forefront of trying to understand humans as sensors. So if you do overdo it, we're going to wind down here very shortly. If you do overdo it, what should you reach for? And it turns out that we have data on that as well. So Gabby Leonard was an undergraduate in our lab, and she was really interested in this question. And so she wanted to know what works the best. So we actually recruited people to bring them into the laboratory, and we gave them the hottest commercial product we could find, beverage we could find. So we went out and we bought Bloody Mary mix. We bought really spicy Bloody Mary mix, and we fed this to people. And then we gave them all sorts of different things to rinse with. We gave them seltzer, beer, water, cola. I will tell you, this was an undergraduate project, so we did non-alcoholic beer, whole milk, skim milk. And what we actually found was that the intuition of folk wisdom is correct, that milk really is the best. But now we actually have quantitative data to show you that milk cuts the burn almost in half right out of the gate relative to nothing. And so this is a great example of how we can use quantitative data to either confirm or what we thought we knew, but really, or sometimes to show that what we thought we knew isn't quite right. And in fact, people for decades have assumed that the reason that milk works best is because of the fat, because the chemical capsaicin that makes hot peppers hot is highly lipophilic. That means it's fat loving. It doesn't like to be in water. It wants to be in fat. And so people have reasoned from first principles, well, if I give you whole milk, then the fat is going to suck up all that capsaicin, and then it's not available to the receptor for you to perceive it as hot. But in fact, what we found is the fat probably matters as well, but we actually took capsaicin and we gave it to people, and we also gave it to them where they either rinsed or as a mixture with casein from milk, just in water, no milk here, just straight casein in water with no fat, or whey protein isolate. And what we find is that both of those dairy proteins actually cut the burn better than plain water. And so this tells us that the ability to sequester that capsaicin and get it off the TRPV1 receptor to cut that burn is not just the fat, but it's probably some combination of the fat and the protein. So as we generalize out here at the end, I just want to talk a little bit about the distinctions between smell and flavor. So much of what we attribute to smell or flavor, you'll hear statistics like 85% of taste is really smell. I don't like that statistic. It's a made up statistic. It varies from food to food. And it also conflates the difference between flavor and taste. And so taste, as used colloquially, means what a scientist would call flavor, whereas taste, narrowly defined by researchers, is the sensations of sweet, sour, salty, bitter, and umami. And so it really turns out that flavor has three different components. It has taste, it has smell, and the smell loss is actually where a lot of people realized that they had COVID early in the pandemic because food, quote unquote, didn't taste right. And it turned out that was mostly due to olfactory loss, but actually some true taste loss as well, which we really had not seen before the COVID pandemic. And then there's also this third part of the chemical touch that comes from thermal receptors like TRPV1 and TRPM8. So they also contribute flavor. So we put all these sensations together and integrate them into this flavor percept that we, all these signals converge to the very front of our brain, just above the eyes, just behind your forehead, but it really is this multimodal input to form that overall flavor sensation. And so humans, I know there were a few questions in the chat about how people differ. So not only do we differ from prior experience, as we see with chili peppers, that you can be high or low based on your prior exposure, but we can also differ in our fundamental biology. So humans have 25 different bitter genes, bitter receptor genes that encode a whole plethora of receptors that can pick up thousands of different compounds that we describe as bitter. Many of them, but not all of them, are toxic. And so the idea is evolutionarily, we didn't want to eat those because they weren't good for us. But it also turns out that at least 4 of these genes in humans have functional variants, where you and I can have the exact same stimulus and have wildly different experiences. So I'm showing you a graph here in the bottom left, where we actually looked at the TAS2R31 gene, and we looked at different variants of that TAS2R31 gene. And what we actually find is the people on the left side of the plot experience more bitterness, less sweetness, and so they like the grapefruit less, where the people on the right actually experience more sweetness and less bitterness, and they like it more. So something as simple as the exact same unsweetened grapefruit, you can feed it to people and they can have a very different experience based on their genetics, and that different perceptual experience can drive differences in liking. Of course, many of you probably have strong opinions about non-nutritive sweeteners, and it turns out that TAS2R31 variants not only explain the bitterness of grapefruit, but they also explain the bitterness of saccharin and acesulfame K. ACE K is commonly used in things like Coke Zero, and saccharin, of course, is ubiquitously available as those pink sweet and low packets at the diner. So I want you to do a little experiment the next time you're at a diner. I want you to taste a sweet and low packet, and then I want you to taste a stevia packet, because it turns out some people experience no bitterness whatsoever from saccharin due to different genetic profiles, and other people experience no bitterness from stevia. But these 2 different genes, TAS2R31 and TAS2R4, are actually not statistically related to each other. So it may be you're a bitter taster for both. It may be that you're a bitter taster for neither, or you're a bitter taster for one, but not the other. So try this little experiment the next time you're at a diner where they have pink and green packets out in front of you. And so what we can see over here on the left, just to bring the point home, is that for the acesulfame K, this gene explains differences, but it doesn't explain differences in rhodioxide A and D, which are chemicals found from the stevia plant. And then over in the top right, what we can see is the people that carry the specific genotype for TAS2R4, some of them experience no bitterness whatsoever and experience it as purely sweet, while other people, based on their genetics, will experience bitterness and very little sweetness from stevia. So there really are big differences across people. And finally, I want to close out by some of the work we've done to try and help people eat more healthful diets. So we did some very extensive modeling work where we were trying to see if we could cut the sugar from chocolate milk, with the idea that chocolate milk is a great way for kids to get calcium and protein. And we wanted to see if we could cut the amount of sugar while still preserving the liking. And so we actually fed 136 adults 22 different chocolate milks, and we did some statistical modeling to see if we could potentiate the sweetness by adding vanillin to them. And then as a follow-up experiment, we ran 142 adults and 160 kids aged 8 to 14, and we just used 5 of the milk samples out of the original 22. And when we did that, I want you to focus in here on the blue box and the red box. So of the different formulations that we used, what you can see at the very top is the overall liking ratings. This numbers along the x-axis, 5, 6, 10.1, 10.2, and 4 refer to the sample numbers. So 10.1 and 10.2 are blind duplicates of each other. So these are the examples of the kind of experimental controls that we use. And so what you can see is, if you look over on the table on the left, you see that sample 4 has about 9% sucrose, where sample 10 only has 4.7% sucrose. In other words, by adding vanillin to the chocolate milk, we can cut the amount of sugar in the chocolate milk literally in half and still get acceptable ratings in terms of the liking. So by using cross-modal interactions between smell and taste, we can preserve that perceived sweetness, cut the sugar in half, and still make a chocolate milk that people like to eat. So the last project that we've done just this last summer was we wanted to see if we could reformulate 10 commonly eaten foods. So we worked with some epidemiologists and some nutritional sciences researchers, and they figured out what are 10 commonly eaten foods that most Americans actually eat to see if we can make an impact in the diet. And so we a professional research chef reformulated these foods to come up with a nutritionally improved version that cut the saturated fat, cut the sodium, or cut the sugar. Not necessarily all three, but it tried to cut at least 2 of those 3 attributes in each recipe. And then we said, okay, so here's what the original recipe tasted like in a blind taste test. Here's what the nutritionally improved recipe tasted like in a blind test. But what happened if we added back herbs and spices to make up for the removal of sodium, sugar, and saturated fat? And we had mixed success. But in blind taste tests, with about 100 people given 3 different recipes for each taste test that we ran, I want to talk you through this top row first. So in the top row, what we can see is the nutritionally improved version is the green one. OG is the original version that has the full fat, full sugar, full salt version. And Fenny, the purple bar, is the flavor enhanced nutritionally improved. So this is lower fat, lower salt, but with herbs and spices added back. And what we actually found is for the chicken and cream sauce, people liked the low fat, low salt, low sugar version with herbs and spices more than the original full fat, full sugar, full salt recipe. The same was true for brownies, and the same was true for chilies. So for these foods, we can actually make a healthier version that actually tastes better than the original full fat, full salt, full sugar version. Then for the middle 4 foods, what you'll notice is that the nutritionally improved version scores a little bit lower than the original version for some of those foods. But we found in these cases that the nutritionally improved version, it wasn't better than the original, but it was at least as good as the original. So it wasn't worse than the original. So now for 7 of these 12 recipes, 10 recipes, we can actually show that we can actually outperform the original version or reach parity. Unfortunately, for 3 of them, we were not successful. So for the last 3, cheese pizza, mac and cheese, and chicken pot pie, adding herbs and spices was not able to make up for the removal of salt and fat in terms of consumer liking. I will point out, though, that these were still acceptable. They were still rated above neutral, but they weren't as good as the original. And putting on both my culinary hat and my food scientist hat, it shouldn't be really surprising that when you take fat out of mac and cheese, take fat out of pizza, or take it out of a cream sauce for that chicken pot pie, that herbs and spices just can't make up for that textural difference. So this is an example of how we can use sensory science to try and help people achieve their dietary goals of eating more healthily while still enjoying their food. So I'll close out here by inviting you to become a taste tester. If you live in the State College area, we would love to have you sign up. That picture on the bottom there, I got here in 2009, so I did not take this picture, but this picture, I believe from the early 1970s, is from the Penn State archives. And these were some of the taste testers. So we have a long tradition of doing sensory evaluation at Penn State. It actually dates all the way back to the 1960s, and the meat slab on the other side of Beaver Stadium from us. And then for 3 decades, it was located in Borland Lab. And then in 2007, we moved over to the current location in Erickson Food Science Building. So if you live in the State College area, if you don't live within the State College area, unfortunately, we can't have you come in for a taste test. But if you live in the State College area and want to sign up, you can use this QR code to take you to a link that will put you in our database, where we'll invite you to come do taste tests on a regular basis. So I will leave that up there instead of going to my final slide, just so that people can scan that. And I'll turn things back over to Bob so that he can ask the questions for the Q&A portion.
Great job, John. Thank you very much. Really interesting presentation. Now it's time for some questions and the opportunity for you to give some answers. Always fun. We had a number of questions that were submitted ahead of time, and you've answered a few of these, but you may want to add a little bit more. So the first question we had prior to the start of the seminar was, how does taste vary in individuals?
Right. So I gave you a bunch of examples already that taste can vary both biologically, innately, due to our genetics. And it turns out that we've known since the 1930s that some people can taste. There's a very specific bitter chemical. You may have had that experience in middle school where your teacher gave you a PTC strip, looked like a little piece of litmus paper that you put on your tongue, and some people could taste it and other people can't. We've known about that phenotype. We've known about that difference, that dimorphism between people since the 1930s. But then with the Human Genome Project in the 1990s, that really exploded. So the specific gene variant responsible for that difference was identified in 2003 when I was a graduate student. And then from there, it's just been off to the races. So as I showed you, there's 25 different bitter genes in humans that you can be. The TAS2R38 gene predicts whether or not you're going to like cruciferous vegetables and alcohol. The TAS2R31 gene predicts whether or not you're going to like grapefruit or saccharin. The TAS2R4 gene predicts whether or not you're going to like stevia. And so we get those differences across people. But we've also learned over the last 20 years that it's not just related to bitter. We also see the same thing for smell. So for example, I have a specific anosmia to androstenone. So I have a mutation in my OR7D4 gene, which is responsible for one of the 400 different odor receptors that humans have. And so I can't smell those musks. So high-end perfumes, totally lost on me. They just don't work for me. Or another one that we've done work on is rotundone. It's responsible for that smell of black, freshly cracked black pepper, but it's also found in wine for Shiraz or Syrah wines. And so if you're in Australia and you're paying $100 a bottle for a high rotundone wine that has that characteristic black pepper smell that you get from a Shiraz, actually, not everyone can smell it because we don't know what the exact gene is, but we know there's big differences across people. And then, of course, I left it implicit in my slides, but it's well known that people differ in whether or not they like cilantro. And so in terms of cilantro, some people experience it as very green, very fresh. Some people instead experience it as very soapy. And so, again, big differences across people, taste, smell, oral touch, all of these can differ. So in fact, the fact that we can even come up with a common language to describe these is kind of amazing in some ways at all.
So you touched on physiologic differences and genetic differences that lead to variance in taste in individuals. This person asks, how do cultural and or religious backgrounds impact food preferences?
That's a great question too. So of course, it's never nature. It's not nature or nurture. It's both. The role of prior experience is huge. And so that some examples of prior experience can include things like chili peppers, like I already talked about. But they can also include things of our familiarity of what we do or don't like. So a classic example of this is that many Americans like root beer. Many don't, but many Americans like root beer. Most Europeans do not like root beer. And most Europeans do not like root beer is because it is something that is very familiar to Americans from childhood, but people don't drink root beer in Europe. And so to them, the closest thing, it reminds them of being of medicine. And so, that's a classic example. Another one is methyl salicylate, which is in wintergreen lifesavers. That smell is used both in lifesavers, but it's also used in toilet cleaning products in some parts of the world. And so that wintergreen lifesaver will smell like toilet bowl cleaner to people from that part of the world. So we do see big cultural differences as well across people.
So here's a question from online. From an anonymous attendee, the question is it ethical to target consumers? What if eating too much of a good thing has an adverse health effect? And they compliment you on the presentation so far.
Thank you. That's a fantastic and provocative question. I think that if we take it to our logical conclusion, we know that nutritional sciences have told people to eat their fruits and vegetables for the last 40 years. And we also know that people's dietary habits are incredibly resilient and at resisting that public health messaging. And so instead of telling people, do eat this, don't eat that, I'm of the philosophy that we should help people eat healthier by giving them food that is healthier, but also that they can enjoy. I don't think we need to bring some sort of aesthetic sense to it where we are avoiding foods because they give us pleasure. And frankly, we live in a capitalist Darwinian marketplace anyway. And so different products are going to appeal to different people. And so if people are going to gravitate to the products that they do or don't like anyway, so, for example, I might really like sriracha, or I might really like Cholula, but I might not like classic Tabasco sauce because for me it's too vinegary. I love the heat, but there's too much vinegar. So this market segmentation is already happening. Just as we go through our daily lives, it can be as simple as my wife and I can make the exact same dish from scratch, and it comes out differently because of how hot we sear the onions, or how liberal we are with the salt shaker while we're cooking it. And so unless we're willing to prescribe that everyone, have the same 3 colors of Soylent, which I don't think we want to do, we're going to have that kind of segmented experience anyway.
Another question that came through is of interest. Many people have talked about food preference and creating foods that taste better in the context of food addiction. Do you believe there is food addiction, or is this a made-up concept?
Food addiction is a tricky one because if you go to the DSM-5 and actually look at how addiction is defined, there's a whole set of criteria that need to be met, and they need to be met in terms of within the judgment of a clinician. And people have been very quick to brand things as food addiction. Unfortunately, I think a lot of the blame goes with the neuroscientists, especially the neuroimagers, who, if we historically looked across the parts of the brain that were hardwired for reward, we know that drugs of abuse hijack the parts of the brain that give us pleasure from food and from sex, because evolutionarily we needed both of those things to survive, or for our species to survive. And so for many years, the framework was, oh, these are really potent activators of this. And so then we're going to go and seek out those drugs of abuse that activates those brain regions. Well, somewhere along the way, the story got flipped by the neuroscientists, because people said, well, this is the part of the brain that responds to cocaine or to opiates or to methamphetamine or whatever substance that they were interested in studying. And then they said, oh, look, food also responds to this. So it really flips the causality around. And, so one of the things that we can think about when we ask the question of if food addiction is real is there dose escalation? Are there psychosocial consequences? And what we find is, no, it's not that eating really pleasurable food makes food not pleasurable anymore. I mean, yes, there's a separate issue of sensory specific satiety that on Thanksgiving, I really can't have thirds of that turkey because now my body is telling me to go eat something else. But across day to day, it's not that food is so pleasurable that now I need to eat twice as much tomorrow to be just as happy. So we don't really see that dose escalation. The other thing is people will use craving or withdrawal as a putative marker for food addiction. Well, guess what? I also have a biological drive for air. So if you were to cut me off from air after about 90 seconds, I'm really going to crave some air, and I need that air to survive. And so is that a craving? Is that a withdrawal? Or is that simply a biological drive to survive? And I think you can make the same argument about food. Now, which food you want to have? Okay, food choice, obviously, people have free will and can choose what they want to eat. But I have to eat some food if I'm going to survive. So I think the entire framework of food addiction is well-intended, but often misinterpreted or misaligned with how human behavior works.
Okay. Question on operationally, the person is curious if you've ever taken into consideration individuals who might be on the autism spectrum and have sensory processing issues. And if so, how do they factor into your data and the feedback that food companies get when they're looking at products?
That is a great question. I have not, up until 4 years ago, I would have told you I'd never worked in a patient population, and that's for the most part true. then with COVID and smell loss, I started to get a little bit more into that end of the field. That said, for the most part, I don't do consumer work in those groups. And so I don't have any direct experience there. There are researchers that are studying sensory processing disorders. Also, another source of picky eating, for example, is many individuals with Down syndrome are very picky about food textures as well. And so there is research going on right now at the Washington State to look at food choices in kids with Downs as it relates to picky eating. So there are researchers that work in that area. I don't work in that area, so I can't comment knowledgeably. Sorry.
So another question on behaviors. And I think this is related to your capsaicin work. How do you test for specific behaviors in panelists, such as sensation seeking and sensitivity to reward?
Right. So there are two different ways you can do this. One of them is you can do prospective stratified recruitment. So we could go and give people a bunch of online surveys ahead of time, and then based on that, recruit specific people to come into the lab. That's not typically what we do, because we're usually studying personality traits that show normal variation across the population. We instead usually just recruit people that can come in and within that 100 people or 200 people find enough natural variability that we don't have to assign people to groups and instead can just look at their data. Now, how we actually assess that, is we use validated tools that come out of the psychology literature. So for example, Arnett's inventory of sensation seeking is a standard tool that is used to measure the construct of sensation seeking. And it was developed as an updated version to an earlier version that back in the 1960s and 1970s was really used to show that sensation seeking predicted all sorts of behaviors. But it also had lots of really dated items. Like one of the items in the original sensation seeking questionnaire was, do you like to hang out with beatniks and other far out groups? I think you would be hard pressed to find 1 in 20 undergraduates who even knew what a beatnik was. So we use more modern tools. So we're also using the sensitivity to reward questionnaire, the risk-taking questionnaire from the personality inventory developed for the DSM-5. So we're using standard tools that exist in the psych literature to try and segment people.
So here's a question sort of related and adjacent. How can sensory science be related to farmers and the future of farming?
So I think that is, again, feeds into our question of market segmentation. If we acknowledge that we're not all the same and that this isn't the 1950s and we're not going to make one kind of cornflakes that appeals to everybody. I think when we're talking about both commodity crops, but also specialty crops, I think we can start talking about growing different crops for different groups. And so what I mean by that is, in general, we don't like bitter things. So we've actually bred broccoli to be much less bitter. We've bred cauliflower to be much less bitter, but we also know that many of those bitter phytonutrients are actually health promoting, ironically. So maybe we've bred some of the benefits out of those things. So you can start to think about, well, maybe my job isn't to appeal to everybody, but my job is to appeal to this smaller niche group. And, obviously, if it's 5% of the population, it's probably not worth targeting. But if it's worth targeting 25% of the population, maybe that's why we see mild, medium, and hots also. Or that's why the celiac community was really thrilled when General Mills decided to certify many of their products that had always been wheat-free, but they actually bothered to jump through the added hoops to actually certify that they were gluten-free so that the celiac community could really trust those products. Candidly, I don't know a whole lot about plant breeding, but I can imagine that you can start to think about, okay, who are we targeting? Just as, on the culinary side, famously in the 1970s, when Alice Water wanted to make some of the ingredients she could not find them commercially. So she actually went out and found farmers to grow the products that she wanted. So I think those kind of partnerships of where we can start to grow products that are meant for specific users and move away from a one-size-fits-all approach probably makes sense. I mean, we already do this with apples, right? I walk into the grocery store and I have a choice of 15 different apple varieties. Well, peaches, when you go in to buy a peach, most people don't even know that there are different peach varieties unless you go to the local farm stand down here on the other side of town, where they'll actually tell you which peach variety there is. So, there might be some messaging and marketing that we can do on the plant science end of things to help people realize, oh, I like this peach more than that peach.
Yep, I think that's fair. A couple of questions that I'll try to bring together. Questions are related to allergies. So related to allergies and food preferences and allergy medicines, antihistamines in food perceptions. And if you wanted to comment on that broad topic.
Yeah, so I think you have two potential different mechanisms there that could be relevant. One of them would be when I take a bunch of antihistamines, it just dries me up in general. And it's not just in my nasal cavity, it's also in my mouth, and you get that cotton mouth. And we know that saliva plays a hugely important role in flavor perception. And in fact, we're only day by day, we're learning more and more about how important saliva is. So I think that when you're on allergy meds that just dry up all your surfaces in the head, potentially they're going to alter what you're experiencing. But on the flip side, one of the major ways that we lose flavor when we have the common cold is we're congested. We're literally plugged up. Technically, it's called a conductive smell loss, meaning that the molecules literally cannot get up to the receptors that sit just right about here, just behind the bridge of your nose, inside your skull at the olfactory epithelium. Those molecules can't get up there when you're congested. So maybe when you go on allergy meds, now your mouth is more dry, but all of a sudden your nose is opened up so you can smell things more. So systematically, which direction it goes do those medicines. And then the final thing that's kind of interesting is, I don't know this is true for the case of antihistamines specifically, but many medicines have weird off tastes to them. And believe it or not, that's actually because they're in your bloodstream, and then they actually get into your tongue is highly innervated. If you've ever bitten your tongue really hard, it bleeds a lot. And so they'll actually diffuse out of the blood supply and hit the taste receptors, not by coming into your mouth from something you've sipped, but actually diffusing up from the tissue through the blood vessels in the tongue. And so that's where a lot of the off tastes of various medications can come from, is actually activating taste receptors because they've infused all your tissues.
John, I want to thank you very much. There are questions we aren't going to be able to hit. Maybe we'll have another opportunity, but I think it's a sign of a good presentation that there were lots of questions and excellent answers. It's time to wrap things up, and I want to thank you again, John, for joining us today in your great presentation. For those of you still in the audience, the next month we hope you'll join us for A Walk in the Woods, a look into the private forests in Pennsylvania and beyond with Allison Muth, director of the James C. Finley Center for Private Forests. She'll discuss the importance of private forests to the Commonwealth, the Eastern U.S., and management opportunities and challenges, as well as research and programs positively impacting their trajectory. I will look forward to seeing you soon. Thanks again, John, for an excellent presentation. And thank you all for joining us today. Have a great afternoon.